think we just need to switch over the slides real quick. So anyway, I'm Katie Bowman from the National Academy of Sciences, so we're obviously really pleased and excited to be hosting this conference, which has been a fantastic experience so far. Thank you so much. And actually, Joan, our committee chair, is going to speak in just a minute about the results of a study that we're very excited to have released in this area, but I thought I would just take 1 second to introduce two other projects at the National Academies that might be of particular interest to this community. So I am not personally doing them, but I would be happy to help connect people who are interested. In particular, I wanted to note a new study that's going to look at Legionella in the water supply. As a lot of you may know, for most of our projects at the National Academies, we really rely a lot on the expertise of committees of you all who are out in the field as active researchers. And so we're currently seeking nominations for that study committee, and I would absolutely encourage you to think about people who would be really good for that project and send them to Laura Ehlers. The email is right there. And then the second project that I thought I just wanted to make sure that the community here knew about is a workshop through our Global Health Unit and our Forum on Microbial Threats that will look at the transmission of infectious diseases in urban environments. So more information about that will be forthcoming in November, with the workshop held in December. I know there's flyers for both of these activities that have been out during the conference, and I just wanted to flag those as forthcoming topics for the community. And so let me now, I guess, turn to Joan Bennett, who is a distinguished professor of plant biology and pathology at Rutgers University, and who was the chair of our committee on microbiomes of the built environment from research to application. And she is going to speak to you all about that project and the results of that study.
Some instructions on.
We seem to be missing, oh, here's just push the green button, there we go.
And brush it here?
No, unfortunately it's over there, which makes it
Okay.
difficult to see.
I've been given 20 minutes to summarize a 296-page project, and we are running late. I will try to move rapidly. This was our charge. If you've been paying attention for the last 2 days, you know that just summarizing what is known is complicated. We're asked to do that. We're also asked to evaluate it and to come up with an agenda for future studies and figure out what was missing. This is a simpler version of what I just said and showed on the earlier slide, that we were asked to find out what we know about the microorganisms in the built environment and how they are changed by our built environment, and then to figure out what we're going to do with that knowledge to make sure that we stay healthy. To quote Jonathan Eisen yesterday, people are everything in doing a project like this. I can't say enough good things about the members of the committee who participated very actively and wrestled to the ground as best we could an enormous body of information. I have a corollary to Jonathan's thing about people. It's not everything. We need money. And the project cost money, and to go forward and do other things, we're going to need money. I thank our funders and, of course, the Sloan Foundation, which has been much of the brainpower behind the whole idea. This is what we did. We had 4 public meetings like this one, in which we brought in experts. Some of you were the speakers at that meeting. So we had a total of 45 speakers, plus our own expertise, plus the literature. And if you want to review all of that, it's available on the web. And then we had to summarize it, put it into a report. And in today trying to figure out how to present this to you, decided I would rather arbitrarily break it down into 4 units, and I recognize that this is a very clumsy classification. And so here, there's the plan. I'm going to start talking about microbes. And something that we didn't do as well as we should have at the beginning, microbiome has become a common word that does not have a common meaning. Among the ecology community, it is a broader idea of everything that's there, perhaps also including all the metabolites. In the molecular biology community, which has really popularized the term, it has more of an omics analysis. And as people come to this area, microbiomes of the built environment, you have to be careful which language the individual is talking. Even scientists don't always talk well to one another. We've been doing this sort of thing for a long time before the omics era, and here's a slide that shows a picture of the old air samplers, which people have been using for years and years to try to get an idea of what kinds of microbes are floating around. What we've added to that with the omics technologies is the ability to learn a lot more than the old culture-based things did. And we're talking about many orders of magnitude. I don't know about you, but when I look at some of these slides with thousands of data points put into different statistical plots, my brain does not always easily summarize what's going on there. And this was part of our challenge. So another part of the challenge, which is not as obvious when looking at these massive data sets, is that we often are finding exactly what we look for. So if you use 16S, you're going to find bacteria, 18S, you're going to find fungi and some other eukaryotes. Depending on how you go about analyzing your data, you will be enriching for one or another of these populations. Sometimes the microbes are living together. We rarely look at things like algae. We hardly know how to begin to study the viruses. And we're not even paying attention. Like in this meeting, we've had very little talk about the things that they make. Mycotoxins. We know there are a lot of bad fungal toxins out there. We know there are a lot of volatiles out there. Microbes are making stuff that may or may not help us or hurt us. We also have variation in where we look. If we just take a residential building, are we looking in the kitchen? Are we looking in the bathroom? How are we looking? Where do we sample? Is it the surface? Is it the air? And then how about the places we don't always look? And not only that, it's all dynamic. It's changing all the time. Each one of these kinds of slides you've seen is usually a snapshot, that moment in time when we looked. I mean, this is complicated stuff. And as I said, these are the kinds of data plots we get that are these snapshots, and that's a lot of information, a lot of information. And we feel good because we can get so much information, but do we understand it? Do we understand its implications? I made this slide early in the genomics era. It's a cover of The Scientist back when I had a hard copy in 2002. That's 15 years ago. We've gotten a lot better at getting information since then. So that's just talking about the omics and the microbiomics. Let's look a little bit at building science, architecture, engineering. When we were trying to define what we meant by buildings and built environments, we decided that we were going to have to narrow our perspective a good bit, and we focused largely on homes, schools, commercial buildings, the place that most of us in Western society spend most of our time. Now and then we would look at the beautiful data that came from specialized environments like hospitals or maybe subways, because that could give us some extra insights. We start running into the same kind of variable parameter overload when we start trying to figure out what part of a building or a building system we are going to analyze the microbiome from, and how is that building used, and what is it composed of, et cetera, et cetera. From the microbiological standpoint, there's a cliche statement that really is true, and the statement is just add water. Water is the most important parameter, and if it overshadows many of the others. It overshadows things like how well the building is maintained, or how it is ventilated, or where the building is situated. And we bring water into buildings because we have to. We need to use toilets and bathe and have heating and cooling systems, etc. And we might be able to build a good system, but eventually things start to leak. And so water gets in from leaks. Yesterday, Ed Jong said you should tell stories. I spent my life studying fungal toxins in foods and only came to my interest in the built environment because I spent most of my professional career on the faculty of Tulane University in New Orleans. And 12 years ago, this is a picture of what Katrina did to my home. And after a lifetime of studying fungi, having the experience of fungi eating my house altered my career. But this shows flooding. I can barely watch the news these days because of Irma and Harvey and Maria, and recognizing that so many people are undergoing what for me was the hardest, except for losing somebody I loved, experience of my life. So there's all that microbes are encroaching, and when the water gets in and they start to grow, they don't only affect us, they affect the building materials in many ways that we don't completely understand. You can form biofilms, they degrade materials. You just heard about this. These are some pictures from Mur. I mean, people are up there in space, and look at the fungi growing on the wiring systems. It was quite horrendous. It's amazing that the spacecraft survived. Microbes can be hidden in parts of the building and then make themselves apparent. We usually see the mold growing on the walls. This is a fungus, a wood-rotting fungus showing up. That shows that there is water hidden in that space. Ventilation's also important, not as important as water, but we've known that for a long time. In fact, we've known some of these things that I'm talking about for a very long time. We now know it in a finer tune. This is a Florence Nightingale picture. Hey, let's keep people healthy by opening the windows. And each one of these factors is very complicated, and we could have a whole talk about any one. More information overload. Okay, that was 2. Now we're on to 3. People. We have discovered through the microbiome study that we shed a lot, we pass gas, we spit, and microbes are being emitted from our bodies all of the time. Jack Gilbert's been very involved in doing some of these studies, and we, I think, underestimated how important we are microbially in our environments. People have tried to do diagrams to illustrate this. This is a very nice one from Jason Pescia's lab. It's talking about the movement of microbes through the building and also on our pets. Here's another attempt to do this kind of three-dimensional diagram that we are actively not only shedding microbes, but we're changing the microbes that are there by our movements around, and as I say, it's very complicated. We differ. Little tiny babies have different microbiomes than old people. Poor people have different living situations than other people. How we eat, how well we keep things clean. Where is the building you're talking about? Is it in Oslo? Is it in Calcutta? Have we used disinfectants? Are there regulations that the government has imposed about how the water is treated before it comes in? Are there building codes that prevent you from doing something? All of this is interacting. And then we have the notion that, hey, we know that buildings can harbor infectious particles. They may also be doing things that are often categorized in a term that does not have an accepted definition of sick building syndrome. This is not the first time the National Academies has approached this, and also the World Health Organization and EPA. And we have this understanding that when buildings get wet, lots of people get sick. Now we come on to putting them all together. How does this infect our health? We know a lot more about what makes us sick than anything else. We know about infectious agents. We understand a fair amount about toxins. And people have been able to categorize each of these cases a little bit. This today and yesterday, we haven't talked much about occupational health. Going back for decades, there has been evidence that certain occupations in which there is a lot of microbiological stuff going on might give you something like cheese worker's lung, or is maple syrup lung on there, or farmer's lung? Yeah. And in our report, we made this nice diagram trying to summarize all of the above about these three things that come together. It's basically a version of what plant pathologists and other pathologists have done for a long time, a simplistic Venn diagram that says, we need the environment, we need the host, that's us, and we need the microbes, and they're all going to come together somehow and maybe make us sick. Our job in this committee was to summarize all this stuff and figure out what the impacts are. And this list, which is in the report, has mostly items about the bad stuff, and then this big, wonderful question mark that's been coming up. Hey, we're finding out that children who don't get exposed to enough microbes young may be sicker than they should be. So we should have known more about this all along, that microbes are also important to us, and we really are just beginning to skim the surface. So there we are. And you figured this out. If you've thought about it at all, everything is complicated. The microbiomes are complex at many levels. The buildings are complex at many. People are complicated, and we move through all of these systems, and they're all interacting dynamically, and there are way more variables than we can handle. And there was a point last spring when I wasn't feeling well, when I just said, there's too much overload. How are we possibly going to make anything sensible? And we have way more questions than we have answers. And I know that when you're the leader, you're supposed to be positive and talk about all the good findings, but I don't want anyone to leave here thinking that we know everything that we should to make recommendations about probiotic microbiomes, or even necessarily what kinds of building standards we should go. We certainly don't know what makes a good microbiome indoors, or we don't even begin to understand what kinds of building characteristics might harbor one, except that we have for many, many years been building buildings that some people find happier than others. We've been intervening all along. Our architecture talk pointed that out. Let's not pretend that we haven't been doing things all along, and we've been intervening in ourselves, the antibiotic revolution, and we clean with disinfectants, and we change the environment by our activities. We're already using a lot of sensors, and if there's a bright spot in this, I think that if people started using more carbon dioxide sensors, we might be able to do a better job of finding out when microbes are growing in large quantities out of control in ways that we hadn't understood before. And I would like to see more sensor research from the engineering community. Here are knowledge gaps. Read the report. I'm not going to go through this particular list, but I want to point out that as we go ahead and make realistic recommendations, that there are going to be trade-offs. I started off using a slide that had the trade-off between energy consumption and maybe a healthy environment. And I have an architect's son, and I switched to this slide because it's considered one of the most beautiful residential buildings, maybe in the world, certainly in the United States, falling water. If you can read the slide there, Mr. Kaufman, who had commissioned this building from Frank Lloyd Wright, pointed out that the building might be called Falling Water, but he thought the nickname should be Rising Mildew, because it has flat roofs, it leaks. The structural problems are even worse, the engineering problems, but there's some serious microbiological problems in this building and a lot of other Frank Lloyd Wright buildings with the flat roofs. Water comes in, mold starts to grow. There are trade-offs. It's a beautiful building, but I don't think it's microbiologically or engineeringly sound. So we came up with a vision for the future anyway. And it was our charge. We did our job, but recognized that we are struggling to put all this together into anything sensible. And so we have these 5 big areas, and then we came down to 12 priorities. And so on the next slides, which are sort of serious slides, I list these 5 priorities with our 5 areas with the 12 priorities. And we need to do a better job of figuring out how the parts fit together, the interrelationships. We should do a better job of the social and behavioral sciences. Engineers and scientists often have a way of looking down on the people who study human behavior, and it's often the single most important factor in a building. We need to look at the way the built environment and our exposures affect human health outcomes. So there are some health-specific hypotheses we can test. We could use model systems. We need to be able to connect the dots, when the health effects happen, how much exposure is associated with it, and then back to this notion of combinations, toxins, other insults, and the microbes. The non-health impacts. The economics is a big driver that scientists don't like to talk about when we are discussing these issues, and often it's the money that talks, not the science. And here we can make a bigger impact. We have to figure out better ways to use our research tools. We can improve the tools we have. I think we could do some good work by having better guidelines for sampling. People are sampling all over the place, and we don't have a standardized protocol. It would be nice to have a data commons. We're looking at different pieces of this enormous universe of trouble, and I think we could use some better modeling tools. And these are things that are all within our grasp as scientists and engineers. And then we need to tell the world, the broader community, what we're doing. And this has become a cliche, improve our communication and engagement activities. It's something we say all the time. We're not always very good at figuring out how to do it, but we could start with our clinical practitioners. Some of the walk-in clinics have some very, very sloppy microbiological methods. Some homes could do a lot just to clean up their microbiological dangers. And then finally, and as somebody who's trained in molecular genetics, microbial molecular genetics, this thing that Ed Young said the other night in his reception address, you can't displace a feeling with a fact. And that sentence works for politics, it works for religion, it works for a lot of stuff. We have spent, the microbiology community, the medical community, the public health community, way over 100 years convincing people that germs are bad and they make us sick. And I found this slide right around before Pasteur. So this is from Punch in the late 1850s, and blaming the River Thames for cholera, scrofula, and diphtheria. Yeah, they were onto something. Our environment is what harbors these germs that make us sick. We are going to have a hard time overcoming this negative image that microbiologists and health practitioners have created for the microbial world. I'm wrapping up. Back to Eisen's lesson. People really are everything. And the report as it exists, all 296 pages of it, would not be as coherent as it is without the wonderful help of the staff. You just saw Katie Bowman. Several of the other people are here. I'm also going to have a shout out for David Butler, who did a beautiful job, and then the consultants who helped with the writing. I thank the people I thanked at the beginning again, our financial sponsors, all the speakers and participants in the meeting who shared their knowledge, and of course the committee members who did the bulk of the original drafting. And finally, Sloan for being the energy behind cohering people to start thinking of the built environment and its microbiome together, and Lynn, who has put this particular meeting together beautifully. It's well orchestrated. Thank you, Lynn. And David Butler came up with a printout of this yesterday that we have not done much publicity for the report, and as of yesterday, there were 3,459 downloads, about half in this country, and then you see the map sprinkled all over the world. We're not the only ones who are aware that the microbiome of the built environment is important to human health and welfare. So that's it. Thank you for listening. I know we're running late, so coffee break? One? Okay. Coffee break. I know you're tired, but hey, it's been a pleasure working with you, and so a particular thank you to those who are still hanging in here.